Good morning, good afternoon, and good evening to everybody, and welcome. My name is Caroline Glover, and I will serve you as your moderator today. Thank you for joining us for our webinar, Make It Right, Best Practices for mRNA Manufacturing. I'm here today with Dr. Narjis El-Ajami and Dr. Shiksha Mantri. Our first speaker is Dr. Shiksha Mantri. She holds a PhD in chemical biology from the University of Oxford, UK, and did her postdoc at ETH Zurich, Switzerland. She supports the top industry players and young startups in the field of RNA delivery and vaccines as the Global Marketing Manager for RNA Solutions. Dr. Narjis El-Ajami is Senior Consultant, Bioprocessing Strategy Operationalization on Novel Modalities and mRNA over EMEA region. She's a molecular microbiologist and bioprocess expert with 10 years experience combining scientific research, process development, and biomanufacturing engineering. Before I turn things over to our two presenters, I'd like to cover 3 housekeeping items. You have multiple application widgets on your screen, and they are all resizable and movable. If you have any questions during the webinar, you can submit them through the Q&A widget. You will also have the opportunity to participate in 3 quick poll questions throughout the session. I encourage you please to take part in these surveys. In case you have some troubleshooting issue, please refresh the page by pressing F5 or try another browser. This will not end the webinar. So that's it from my side. It's my pleasure to turn things over to Shiksha first, and after that to Narjis. Then we'll have a 15-minute Q&A.
Hi everyone, my name is Shiksha Mantri. The reason why we have chosen this topic as the webinar is because mRNA has taken center stage. mRNA vaccines were the first COVID-19 vaccines to be approved, and there's tremendous potential to do much more. So in this webinar, we would be covering what is the power of mRNA. Then we would be going into the factors that impact mRNA performance. Then Narjis would be covering how to make the right mRNA, and in the end, we would be covering the key takeaway messages. So let's get started. mRNA is not a new molecule in itself. It was first discovered in 1961. Fast forward 60 years, and we have the first mRNA vaccines approved. But the record speed approval of these vaccines was based on 60 years of research and development. Now, to really look into what mRNA can do, we have to look what it does inside the cell. In cells, all proteins are made through mRNA. This means that we can introduce foreign mRNA and produce new proteins in the cell. And this in itself has huge implications. For example, one could use it to manufacture antigens in the cell, and thereby use it for vaccines. The biggest example, COVID-19 vaccines. But the mRNA could also be used, for example, for cancer vaccines. One could also deliver the mRNA of enzymes for enzyme replacement therapy, or therapeutic antibodies, or proteins for gene editing. And in the end, one huge potential for mRNA lies in personalized medicine and also for ex vivo gene therapy. As mentioned, mRNA was being actively researched for a variety of therapeutic indications before the COVID-19 pandemic hit. But then the approval of the mRNA vaccines for COVID-19 was an excellent proof of concept, and we have seen a huge increase in the number of preclinical and clinical programs since last February. So to discuss some numbers, in February 2020, there were 129 programs altogether for mRNA. Fast forward 1 year, in April 2021, there were more than 220 programs using mRNA. And these were not just COVID-focused. They were also focusing on other infectious diseases. There was a big chunk of research being focused on mRNA for oncology, and there were a variety of other indications being researched as well. Now let's discuss some factors that impact the mRNA performance. Each step going into mRNA manufacturing and formulation can critically affect the activity of the final drug product. A typical manufacturing process looks like the following. Plasmid DNA is linearized, which can then act as a template for the in vitro transcription reaction, where the DNA is then converted into mRNA. The mRNA then has to be purified using multiple steps to produce pure mRNA. This is typically followed by formulation steps using lipids or some other excipients, which is once again then followed by purification steps to yield the final drug product. So we have distilled down the factors that can impact the mRNA performance into 3 pillars. First one is the mRNA identity itself. The second one is the purity of the mRNA. And the third one is the formulation parameters. And we'll be discussing through these key factors in more detail in the next following slides.
Hey Shiksha, now I have a first poll question for our audience. Question is, are you working on, involved in mRNA therapeutics development? Yes, actively in clinical trials. Yes, doing preclinical trials, or considering to start, or no, not working with mRNA. And I'll give you a few moments to vote. Results are coming here. Thanks for voting. Now back to you, Shiksha.
Let's start with factor number 1, the mRNA identity itself. Here we discuss the main structural elements of mRNA and how they are important for the stability and the performance of the molecule. At the 5' end of the mRNA, one would usually cap the mRNA. The cap is for the protection of mRNA from degradation by exonucleases. The cap also plays an indispensable role during translation because the eukaryotic initiation factor binds to the cap. Depending on the cap structure used, it can also prevent innate immune sensors from recognizing the mRNA. The next bit is the 5' and the 3' UTR regions. UTR is untranslated regions that flank before and after the coding sequence for the protein. The UTR regions affect the protein expression and the mRNA stability. Then you have the coding sequence. This is the part that encodes for the protein that is desired. We'll shortly discuss the 3' UTR as well. And in the end, you have a poly A tail. The length of the poly A tail affects the stability and the translational efficiency of the mRNA. Let's begin with the cap structures. The cap structures typically available are cap0 analogs or cap1 analogs. So ARCA is a cap0 analog that can be added to the mRNA at the same time as the IVT reaction. However, it has some certain drawbacks as well, such as that the capping efficiency is limited. This means that a phosphatase treatment may be required after the IVT reaction to remove the 5' phosphate from the uncapped mRNA. The other option typically available is a cap1 structure. Cap1 structure can be achieved through the Vaccinia capping system. This process is typically done after the IVT step. However, it has multiple advantages as well, such as having no IP restrictions and a very high capping efficiency. Let's now discuss the ORF and the untranslated regions. The ORF, or the open reading frame, is the coding sequence for the desired protein. For the coding sequence, one needs an optimized codon sequence, and here one also needs to determine if and which modified bases to use in the sequence. The UTR, or the untranslated regions, are essentially regulatory sequence elements. These can affect the mRNA trafficking and translation, among other features. For the UTR regions, any reliable manufacturer should be able to advise you on the sequences to use. And the last, but definitely not the least important feature of the mRNA identity is the poly A tail. The length of the poly A tail affects the stability, translation, and the performance activity of the mRNA. An inhomogeneous poly A tail length would imply an unpredictable or a not optimal mRNA activity. In a typical mRNA manufacturing process, one usually has to start with large amounts of plasmid DNA, which is then linearized. And this step in itself can have major implications on the mRNA that is produced. So in the next couple of slides, we'll be going through some of the drawbacks that this approach may have. So for considering the drawbacks of a standard mRNA manufacturing process, one has to keep in mind that the plasmid DNA, the DNA sequence itself, is acting as the template for the mRNA sequence. So if your DNA sequence is not perfect, the resulting mRNA would also not be perfect. So when producing the plasmid in microorganisms, let's say E. coli, there could be replication slippage during the replication of the plasmid DNA, and this could lead to truncation of the homopolymeric T120 region in the DNA. And the effects of replication slippage means that you have a randomized mixture of variable sequence lengths of the poly A tail as the template for the IVT. And this means that the mRNA you receive at the end would also have variable poly A tail lengths. The second potential pitfall in a standard mRNA manufacturing process is that the plasmid DNA needs to be linearized before the IVT reaction. The linearization step of the DNA is usually done with DNA cleavage enzymes, but the problem is that there could be incomplete plasmid DNA cleavage, and this could be very difficult to trace. Next, we'll show some data on this. Here you see some data where plasmid DNA was digested with 10 times more enzyme than usually used, followed by a fragment analyzer run. And it was seen that even after overnight digestion with 10-fold more enzyme, there was still some undigested plasmid DNA left. During the IVT reaction, the T7 RNA polymerase used is an extremely processive enzyme. And if you have traces of plasmid DNA left that are not cleaved, it will continue to transcribe around the circular template multiple times before dissociating. And what this essentially means is that one could have very long and poorly detectable trailing mRNA products that could impact the activity of your mRNA. Another limitation of the standard process is that the plasmid DNA could have contaminating DNA from the host DNA, such as E. coli DNA. And the contaminating DNA can interfere with the mRNA manufacturing process. It can also be left behind in the final formulation, thereby affecting your performance of the mRNA. And the result of this is essentially unexpected in vivo activity and potentially also inflammatory response. In a standard mRNA manufacturing process, one requires large amounts of plasmid DNA to begin with. Estimates go between 100 and 150 milligrams of plasmid DNA required for every gram of mRNA to be produced. This is an important supply chain consideration to keep in mind. Earlier this year, Merck acquired Amtech. Amtech is a nucleic acid manufacturing company which is based in Hamburg, Germany. For mRNA, Amtech has developed a unique PCR-based workflow that eliminates all the problems that we discussed associated with the standard mRNA manufacturing process. So first of all, the plasmid DNA required for Amtech's process doesn't need to have an encoded poly-A region. This implies that the template that is used for the IVT is highly homogeneous and has a highly homogeneous poly-A tail. Secondly, there is no linearization step required. This means no, if at all, little plasmid DNA is left over when going into the IVT step. And thirdly, the amount of plasmid DNA required for Amtech's process is approximately 10,000 times less than what is required for a standard process. On the right, you see the template that would go into the IVT reaction that was made using Amtech's PCR-based workflow. With Amtech's PCR-based workflow, one can achieve a highly homogeneous and pure template for the IVT reaction. What this means for the resulting mRNA is that the mRNA has a highly consistent poly-A tail region, and this has a direct impact on its performance as well. Amtech's PCR-based workflow is highly reproducible. This implies that the mRNA coming out of this process has highly consistent activity as well. Now, with regards to the performance of mRNA coming from Amtech, in the next slides, we'll be showing you some data. Here, we would like to share some data that provides evidence for the high activity of mRNA coming from Amtech. In this study, 3 batches of mRNA from Amtech were compared to a commercial standard. It was observed that the 3 batches from Amtech were statistically equivalent. The experimenters also saw that the mRNA from Amtech was 2 times more active than the industry standard, and the Amtech mRNA also led to lower plasma cytokine levels after 4 hours of in vivo injection. Switching gears a bit, another very important watch out while doing mRNA manufacturing or formulation is to watch out for endonucleases. We have a poll question for you for that.
Let's go to the audience for a second poll question. Question is, in which step of the process is the tested for endonucleases claim important for you? For all steps, IVT chemicals, purification consumables, formulation chemicals, or not important? We give you 10 seconds to answer this question. And the results are shown here. Thanks again for voting. We really do appreciate your feedback in this very important topic. Now we return back to you, Shiksha.
In all steps of mRNA manufacturing and formulation, starting from lysis of the cells for plasmid DNA production to the chromatography steps, to the encapsulation steps, and the final formulation, there are a variety of raw materials used. The raw materials used vary from solvents to buffers to salts and stabilizers, and all of these need to be endonuclease free because endonucleases can completely chew up your mRNA. So Merck has several different chemicals as improved expert raw materials that have been tested for endonuclease activity and have very specified low bioburden levels. They comply to the IPEC PQG GMP compliance as well. And to know more information, please do reach out to us.
Thank you, Shiksha. My name is Nargis El-Hajami, and I have the pleasure to take over here to present you some other manufacturing key considerations to help you make the right mRNA. After you make sure that you have the right mRNA identity via the right DNA template, it is crucial to make sure that your mRNA is efficiently purified from product and process contaminants, and that the final mRNA product is properly formulated into the right delivery system. The purity of the mRNA is a crucial determinant of yield and efficiency. Impurities removal reduces innate immune responses and results in significantly higher levels of reporter protein expression. As you could see earlier, depending on the origin of your DNA template, the type and level of your contaminant would be different. Three technologies are mostly used for mRNA purification: chromatography, tangential flow filtration, and normal flow filtration, usually consisting on a sterile filtration at the end of the purification step and, if applicable, at the end of the formulation step. Chrom and TFF have both high impurity removal efficiency, and one or both would be needed after each enzymatic reaction step, like plasmid DNA dimerization if using standard plasmid from bacterial process, the in vitro transcription reaction, or enzymatic capping. However, you need to take into consideration existing patents around these technologies for mRNA purification when designing and developing your own process. Impurities can be enzymes, including restriction enzyme used for plasmid linearization, polymerases, enzymatic capping enzymes, DSA, residual solvents, elemental impurities, residual DNA templates, small oligonucleotide impurities that can be due to abortive initiation events by the RNA polymerases, double-stranded RNA generated by self-complementary 3' extension, RNA-DNA hybrids, or secondary RNA structures, known as hairpin contaminants. Such impurities can induce strong inflammatory reaction and side effects and need to be closely controlled prior product releasing. Now, what are the key process considerations that can impact the mRNA purity and manufacturing process? It is important to start right to finish right. For this, you want to consider relying on a good process development expertise to quickly design an efficient, scalable process for your mRNA. This would also help you build the right process parameters required for your mRNA in terms of, for example, temperatures, pressures, flow rates, but also the choice of media, materials, and chemicals that are required. It is important to take into consideration the scale-up of your process while designing it at small scale. For example, most of the lab-scale processes use solvent extraction and precipitation steps for the mRNA purification. These methods are hard to scale up and use hazardous solvents that are not suitable for GMP conditions. These steps can be replaced by some filtration steps, TFF, and chromatography, as we've seen earlier, for a more efficient GMP manufacturing process. It is crucial to assure that any raw material, solution, or equipment used in your process are free from endonucleases. If your final product is a large mRNA complex, you want to consider the alternatives for sterilizing your product in advance and designing the best ways to do it. Finally, it is also very important to consider the stability of your drug and plan for storage, cold chain, and shipment logistics beforehand. If we summarize, it goes without saying that making the right mRNA therapeutic would require having the right mRNA manufacturing process with the right and efficient purification steps. If you would like to learn more about each step of mRNA production process and the advantages and disadvantages of each technology, I invite you to watch my previous webinar Unlocking the Potential of mRNA Vaccines and Therapeutics. You can find the link attached here. Now, let's move to the third critical quality attribute, which is the delivery system. Having the right delivery system able to efficiently and safely deliver the mRNA into the cytoplasm where protein translation takes place is also what drives mRNA efficiency. The most advanced class of delivery systems are complexes of lipids and polymers, known as lipoplexes and polyplexes, respectively. For mRNA delivery processes specifically, the lipid nanoparticle, known as LNP, is one of the most used ones. Now, how do we formulate the purified mRNA into these magical LNPs? The most used technique is the solvent injection technique. The lipids are dissolved in a solvent, usually ethanol, and then it's rapidly mixed in an aqueous low pH buffer containing the mRNA to create these LNPs. The low pH buffer of the mix is then exchanged by a diafiltration into a neutral buffer and ultrafiltration to concentrate the particles. A stable lipid nanoparticle consists of 4 different lipids, allowing the mRNA to be carried in the core of the LNP and being protected from degradation. The 4 groups of lipids impacting mRNA stability and efficiency are, first, the ionizable cationic lipids that will bind to the negatively charged mRNA and then fuse to the cell membrane to release mRNAs in the cytoplasm through ionization. The second group is natural phospholipids, also called structural helper lipids, that would support the nanoparticles to form a lipid bilayer structure. Third type of lipids is the famous cholesterol that is used as a stabilizer to increase LNP stability. And finally, the PEG lipids that provide stability and also can extend the half-life of LNP complex. As the mRNA is formulated with the 4 lipids, the mRNA is carried in the core of the LNP, which can be protected from degradation. We have a very broad portfolio of the 4 types of lipids, and we also have custom manufacturing to make custom and special lipids. And if you want to learn more about mRNA formulation and how to make the right choice of lipids, you can refer to Shiksha's webinar, Key to Successful Formulation Development for Lipid-Based RNA Delivery. You can find the link attached here as well. Now, let's look at the key drivers for a successful mRNA manufacturing. First of all, it goes without saying that having the right production process is crucial. The development and manufacturing process of mRNA are comparatively simple, scalable, more straightforward, and extremely rapid, and one should consider robust, efficient, and fast process steps and technologies when building his production process. For this, having the right expertise and experts is important. It is a big strength today to have the right mRNA expertise from R&D to process development and manufacturing optimization. The third success driver is working with the right partner, not only for assuring the right supplies, but one would also consider the right strategy in terms of outsourcing some activities to CDMO's partner and collaborating with institutes to accelerate and acquire processes. Finally, one crucial element for the success of any therapeutic production is having the right facility design. For such innovative technology like mRNA, it is important to consider flexibility and next-gen solutions when drawing your facility design. Before going further, we have a last poll question for you.
Now we have a latest poll question for our audience. What is your biggest bottleneck toward an efficient mRNA production? The production process, the expertise, the partnership, or the facility design? Take some few time to vote. Thanks for voting. Results are here. and giving certainly interesting insights for our audience as well. Now I'm turning it over to you, Nargis. Go ahead.
Thank you, Carol. I would like to dedicate this last part of the webinar to mRNA facility design and how to make it successful. Let's look at the trends today for the facility of the future. Next generation processing like single-use solutions, closed systems, continuous and intensified processes are changing paradigms in space allocation and classification, but also in terms of time, footprint, and cost savings. In parallel to that, we are seeing increased automation and digitalization in all aspects of a facility, including elements of process, quality, procurement and distribution systems, providing a more integrated manufacturing and supply chain. This would allow an automated management of the planning and execution of an mRNA vaccine, for example, from design and scale-up to manufacturing and distribution to the regulated post-market surveillance of the product. Our company is the first supplier to converge advanced process technologies with software, automation, and analytics into a first-of-its-kind digital ecosystem, the Bio4C software suite. This transformative software suite will allow users to look across the entire manufacturing process versus individual operational units, giving biomanufacturers complete process control and deep insights, bringing Bioprocessing 4.0 to the here and now. We have also launched two other Bio4C products. The Bio4C Process Pad, that is a data visualization, analytics, and process monitoring platform that enables bioprocess lifecycle management, reporting, investigations, and continued process verification. Third product is the Bio4C Orchestrator software. This one allows you to monitor and visualize your bioprocess from anywhere. The third trend is related to flexible design. Understanding mRNA manufacturing approaches with both product and process flexibility is now a requirement. mRNA technology provides the ability to easily establish flexible manufacturing processing, allowing for the rapid changeover between either existing product campaigns or new development candidates with minor process adaptation, which allows again changing and having a significant time and cost savings. Modular and modular prefabricated facility designs have become a regular consideration for many projects. They offer increased flexibility and enhance environmental sustainability and can facilitate validation and regulatory processes. They provide reduced footprints, time to market, operating expenses, and service requirements. These deployable units can allow a fast capacity increase when needed and can be shipped to locations where manufacturing is needed. When designing your mRNA manufacturing facility, there are some questions that must be addressed, like what are the process operations and the level of automation that should be included for my facility? What is the level of segregation required, and what is the best strategy for separating the unit operations? Which production environment is required for unit operation of mRNA manufacturing process, such as containment and air turnover? Where will the facility be located, and are there any specific local requirements, space limitations, or existing buildings that need to be taken into consideration for the design? Is your facility designed to assure reliable production? And does it enable meeting future regulatory expectations or technical change without major shutdown? Do the facility design and construction support rapid deployment and minimize the time to market? How can you increase safety and avoid risks within your facility? Once you have addressed all these points, you want to make sure that your facility design fits to the business strategy as well as the process. Those need to be clearly defined in terms of product types and size, like are you producing self-amplifying mRNA or non-replicating mRNA, the volumes, and also the yields expected, and so on. On the other hand, the fit to the process should highlight the level of flexibility that we want to achieve in order to choose the right technologies from single-use to hybrid. All this information and anticipations are crucial to design the right facility and to do things the right way at first time. I wanted to finish this part by sharing with you a case study that we recently finalized with our customer in KSA, where we worked on revamping an existing facility to make a new conceptual facility design for an mRNA CDMO facility. This study took into consideration all the flexibility, agility, operational efficiency, and cost effectiveness that an mRNA conceptual facility design should have. The facility included a plasmid DNA production unit, and based on provided inputs, we could provide different deliverables, including detailed facility layouts with process ways, people and material flow, room classification, pressure cascade, process flow diagrams, equipment lists, CAPEX and OPEX, 3D layouts and plans, in addition to optimization proposal and other outputs that I will be glad to share with you in our next webinar that will be dedicated to facility design, so stay tuned. Now, before closing this webinar, I have some key takeaways for you. I hope we could give you a good overview and good flavor of the different key elements to consider in order to make the right mRNA therapeutic. mRNA technology holds great features in terms of manufacturing rapidity, simplicity, and efficiency. Three major critical attributes impact the mRNA efficiency: the mRNA identity itself, where the choice of DNA template is impactful, the purity of the mRNA, and the mRNA formulation or the delivery system. In alignment with the need for speed challenge that mRNA technology showed the ability to tackle, it makes sense to consider advanced technologies, novel processing concepts, and next-generation manufacturing to design the facility of the future for mRNA production. Finally, this pandemic allowed us to witness the power of choosing the right partner to accelerate success. Choosing the right partner and starting right with a robust conceptual design are key to make it right. I would like to end up with an overview of how our company is supporting this booming involvement of mRNA field. Through our different divisions, we have a broad range of integrated solutions to help you make, purify, and formulate your mRNA products and assure robustness and quality for such process. Our integrated offerings go from bioprocessing devices for each specific process step to a wide range of raw material, excipients, chemicals, and lipids to design an efficient mRNA delivery system with high quality. We are also able to answer to the main challenges raised by the single-use and close processing consideration with our single-use kits and sampling devices, as well as the innovative solution for closed system processing. Our offering also includes testing services to guarantee consistency and ensuring highest level of product quality and safety, in addition to engineering services like conceptual facility design studies and consultancy, as well as process development support. And with this, I would like to thank you very much for attending this webinar, and I look forward to taking your questions.
Thank you for viewing. Thank you both Nargis and Shiksha for this great presentation. Now it's time to answer a few questions that have come in from our audience. So we have enough time, I think, and we'll try to get through all of these questions, but if we run out of time, we will respond to you individually later. Now you're going to start answering questions that have come in. And I have a first question for you, Shiksha. That is, what is the size limit for PCR approach by Amtech?
Okay. Thank you, Carol. So the question is, what is the size limit for PCR approach by Amtech? We can do anything that's between 200 nucleotides to 13,000 nucleotides with this approach. The 13,000 nucleotides, it's not a hard limit. So if your size of the mRNA desired is longer than that, then please, you could still reach out to us. I hope that answers your question.
Okay, thank you, Shiksha. Thank you very much. Let's move to the second question, and it will be for you, Nargis. The question is the purity of pDNA needed to make mRNA?
Is the purity of pDNA needed for mRNA production? So the answer is yes. And actually, the purity of plasmid DNA is very important and can impact the mRNA purification process, because if you have some impurities that are coming from your plasmid DNA process, so you want to make sure you are getting rid of it. And some contaminants from plasmid DNA may come from E. coli, so like endotoxins and so on. So you want to make sure you are getting rid of all those contaminants. And again, this will impact your mRNA process specifically, because you may need more intensified purification step at that level. So yeah, the purity of plasmid DNA is very important, and the grade as well of the plasmid DNA we'll be using for mRNA synthesis would be important as well. So you want to make sure you are choosing a good grade for your plasmid DNA purity, and being aware of all the contaminants that are there.
Thank you, Nargis. And I think we have an available on-demand webinar broadcasted quite recently about this topic, and more precisely about data-driven strategies and consideration for scalable purification of plasmid DNA. So for those who are interested, just reach us out. Shiksha, I'm going to have a question for you. What is the key process step that determines entrapment efficacy of mRNA?
Okay. So regarding the entrapment efficiency of mRNA, there are actually several parameters that determine that. And we had a webinar last year where we actually covered this topic in great detail. So please feel to have a look at that as well. But in short, like even the choice of lipids has a big impact. So depending on which ionizable lipid is used, what is the ratio between the ionizable lipid and the mRNA in terms of the positive to negative charges, so N to P ratio, determines the encapsulation efficiency. The encapsulation method used in itself can have a big impact on how much mRNA is being entrapped. Then there are several other factors as well. For example, if your purification process is too harsh, then you can actually lose the lipid nanoparticles and in the end lose the mRNA, thereby reducing the encapsulation efficiency. Then there are also other softer factors which are easy to ignore but extremely important, and those would be, for example, the purity of lipids that you're using, because if you have lipids that have impurities such as free fatty acids or so, then it's very easy to make a lipid nanoparticle which is actually leaky, and this has an impact on the encapsulation efficiency. So I would recommend do have a look at this webinar as well, and please feel free to contact us if you require more information.
Okay, thank you, Shiksha. I think it's an accurate answer, and I hope this help our audience. We have another question that is, how big size of bioreactor is used to the common mRNA process for production? And Nargis, I think you can take this question. If you'd like me to repeat.
Yes, can you please repeat, Carol?
Yes. How big size of bioreactor is used to the common mRNA process for production?
Okay. So first of all, usually there is no cell production for mRNA. It's fully synthetic, cell-free, so no bioreactor is needed, no cell culture. It's more about mixers and reactors. So if the question is about that, it depends on the doses that you want to achieve. And the doses would also depend on what kind of mRNA you are talking about. So you need to know that today mostly used mRNAs for vaccine or therapeutics are either non-replicating or self-amplifying mRNAs. The difference between the two is that the non-replicating mRNA is, let's say, the normal form of mRNA, where we show the sequence here during the presentation. And self-amplifying mRNAs are mRNA with a sequence that has a gene, a replicate, that would allow a self-amplification of mRNA when it's inside the cytoplasm. And why this is like a new form of mRNA or next generation? It is longer in terms of sequence, but it would allow to use much lower doses. Like it can go up to 200 times lower doses in terms of micrograms per dose. So depending on the number of doses that would be required, you would determine the size of your reactor and what are the volumes that would be needed. But today, for example, what we see with non-replicating mRNA, which are for example, the validated vaccines, the batches can be 2,000 liters, and there are other companies that have vaccines where batch volume is around 50 liters. And yeah, so this is for the mRNA process specifically.
Okay, thank you. Thank you. So I'm shifting to Shiksha about this topic about mRNA process, because we have someone requiring more details, Shiksha, on Amtech's mRNA manufacturing process.
Okay.
Can you advise us?
Yes. So actually, I see in the Q&A that there is a similar question on this topic, which is about the homogeneous poly-A tailing of mRNA. Is it enzymatic or chemical? So we can answer both questions at the same time. So essentially, to just share some more details on Amtech's mRNA manufacturing process, this process was actually developed by the founders of Amtech. They developed a unique PCR-based workflow, and they perfected it over 15 years of Amtech's existence. So the template for the IVT is not the plasmid itself. This implies that the plasmid that Amtech would use doesn't need to have an encoded poly-A tail region. And this poly-A encoding region is actually introduced into the template for the IVT reaction through the PCR-based workflow. And this is the reason why the resulting template has a highly defined and homogeneous poly-A tail. Beyond this, I'm afraid I cannot share more details about the PCR-based workflow, but I hope this answers your question.
Thank you. We have a lot of questions, so trying to give some priorities to them. So, and shifting from Shiksha to Nargis. So now it's a question for you, Nargis. The question is, how do you avoid endonuclease contamination brought in by the operator itself despite closed process design?
So obviously to make sure that you're not having any endonuclease in the process, you need to use, as I mentioned, endonuclease-free products, and anything that would be in contact with your product, with your mRNA, should be RNase-free. And in terms of the operator that will be handling the process, of course, you need to make sure, and he needs to make sure that he doesn't contact the product anyways, even the buffers when getting prepared. So all the steps need to be carefully made in a way that the operator would not be in contact with any of the solutions or materials that are in contact with the mRNA. And this is exactly what can be avoided when working in closed system and minimizing and automation as well, to minimize any operator intervention to avoid the risk of losing or of contaminating the products with mRNAs that may come from the operator.
Okay, thank you. I'm checking if I see some similar questions around the same topic, but there's no. So, and in which, a question is, how does type of therapy and module vary the process? Any idea which one of you, if someone of you would like to answer this question? How does type of therapy and, sorry? Can you hear me? So my question is, how does type of therapy and module vary the process?
This type of therapy varies the process.
Okay. There's another question that is, what is major concern of multiple product production in terms of product change over? And I'm not sure I catch this question well. Or would you prefer to answer to this question? How small a footprint do you think mRNA will enable us to get and help provide personalized therapies? Can someone answer? We can't hear you.
So the question is about the footprints, and it talks about personalized therapies, if I got it right.
Yeah.
So the footprint, how small a footprint? I think you are talking about the facility. So I don't know if the person can clarify a little bit more the question. But in terms of footprint, again, if we imagine the facility, it is much smaller compared to traditional facilities that we are used to in terms of vaccines or therapeutics, simply because we don't have this upstream parts and downstream separated. We don't have this, especially the upstream parts where we do need bioreactors and culture media and so on. So the segregation usually is made on the function where we have like the media preparation area, and then if we don't have the plasmid DNA part, like only the in vitro transcriptional parts, and if we are having the linearization, it can be like really done at the same place at the mRNA, and then the formulation, you have the choice of having it at the same area or having a segregation. You can have formulation parts for lipids separate or not, so you have a lot of flexibility in that sense. But in terms of footprints, definitely smaller, and how it can serve personalized therapies. So a lot of work is ongoing to treat cancers and some other application therapies for using mRNA, because it really opens the door to much simpler processes in terms of manufacturing, production, and also facility, ease of facility and adaptation to those novel designs that we can have.
Okay.
I hope that's the question, and then
I think so, yes. And what are the most important measures for providing a safe, nucleus-free clean room environment? Is a nucleus monitoring possible and recommended? I don't know if this question is clear for you. I can repeat. What are the most important measures for providing a safe, nucleus-free, clean room environment?
So again, having a nuclease-free environment is really about taking all the measures of minimizing the operator's contact with all the equipment. So it would be it's going to be a great C environment with applying all the safety measures to avoid any contamination or contact. That's the biggest caution that are taken when handling mRNA process.
Yeah, and similar question is, are the chemicals provided from our company free of endonucleases?
So we do have a broad list of chemicals that are endonuclease free, and we are working on new ones that will be coming as well. But we already have a very broad portfolio of chemicals that have the claim of being nuclease free and that are used for mRNA synthesis.
Thank you, Narcisse. I have maybe a more specific and technical question, if you can answer. What is the maximum scale in liters recommended for in vitro translation step? What is the maximum scale? Sorry?
The maximum scale, yeah, and I got the question previously about the bioreactor scale or the reactor scale.
Yeah.
So the maximum we have seen is, as I said, it's 2,000 up to 2,000 liters, yeah. And again, there are also processes, and I believe you have a better handling, let's say, of lower volumes like 50 liter or 200, but we do see, and there is processes with 2,000 liters where the in vitro transcription is made, and but for we have seen data stating, and I've seen publications stating that you can also split those volumes. For example, if you need to achieve 2,000 liter, you can split into 4 different bioreactor or 500 to have a better monitoring, and then you pull things together at the end of that step. But we also have seen mixers of 2,000 liter handling the in vitro transcriptional step.
Thank you, Narcisse. I'm shifting slightly to, I have several questions around LNP. So the 2-3 coming questions are for you, Shiksha. And you can answer. What is a common LNP batch size for production, and how much mRNA can you bring in? This is first question. There's also a question around if during LNP formulating process, what volume of ethanol to lipids, what volume of acetic acid to mRNA, if I want to make 20 liters of final encapsulating product. So these are really accurate, specific questions. And if you can end with that, because then we'll be running out of time, Shiksha. So you have 2 minutes.
So, Carol, these questions regarding the LNP batch sizes and the facility, essentially, they are highly variable, depends on the application, and it's super difficult to answer them straight away. However, we could take a question regarding mRNA. For example, there's one which asks how much pDNA is required to make mRNA. So in a standard process, one would use approximately 100 milligrams to 150 milligrams of plasmid DNA to make 1 gram of mRNA. And in the AMTEC process, since it's a PCR-based workflow, we would require 10,000 times less plasmid DNA to make the same amount of mRNA.
Okay, and to be honest, we have a lot of questions, so but we're arriving to the end of this webinar, so I'm closing this live Q&A session. I thank you very much, the audience, for all their questions. And this webinar will be available on demand on our website soon. You can access it using the same link than the one you're using now. And for all these unanswered questions, you will receive an individual email soon from our experts in the coming day. So again, I would like to thank you, Shiksha and Nargis, for today's such great presentation, and a big thank you to our audience for joining us. And some more will follow up soon. Have a great day.